All right, Chapter 8, Cellular Respiration. This is a continuation of our energy unit. This should finalize our chapter on energy before we move into genetics and molecular biology. There are 5 sections in this chapter. First, I'm going to look at an overview of cellular respiration. Second part, 8.2, we'll look at outside the mitochondria and the process of glycolysis. And then we'll look at in 8.3 the process of fermentation, which is also outside the mitochondria. And then we'll go inside for section 8.4 into the mitochondria, where we'll see cellular respiration take place and look at metabolism in general. So overall, cell respiration, it is that cellular process that breaks down nutrient molecules produced by photosynthesizers with the production of ATP there. It is a catabolic reaction. It consumes oxygen, produces CO2. That is if it is a true aerobic process, meaning that it requires oxygen gas. Cellular respiration itself is aerobic. It also usually involves a complete breakdown of glucose to form carbon dioxide and water as byproducts. And remember that carbon dioxide and water are the reactants to photosynthesis. So energy is extracted from glucose or harnessed from glucose. It's released in a step-by-step processes that uses the help of enzymes to produce ATP. Like photosynthesis, it is a series of oxidation reduction reactions, and we're going to have two electron carrier molecules, NAD plus and FAD, that are going to serve as coenzymes in this cellular process. So if you look, the two things that are being oxidized in cellular respiration are glucose and carbon dioxide, and things being reduced are going to be oxygen gas and water. So cellular respiration itself, if we look into the typical eukaryotic cell, and this happens to be the eukaryotic cell of a leopard, you do see what the mitochondria looks like. The mitochondria is, like the chloroplast, considered to be once a free living organism that has been brought into the eukaryotic cell. Therefore, due to that endosymbiotic theory, endosymbiosis, as it was brought into, engulfed into what we now would consider the eukaryotic cell, we have that second membrane. So it's a double membrane bound organelle, and that inner membrane has folds, and the inner membrane is folded, and those folds are called cristae, and the cristae are going to increase the surface area for the reactions. You also have the area between the inner and outer membrane called the intermembrane space, and on the very inside of the mitochondria, within the inner membrane, is your mitochondrial matrix. So the two coenzymes that play a role in cellular respiration are NAD plus and FAD. NAD plus stands for nicotinamide adenine dinucleotide. It's a coenzyme of oxidation reduction, and it is oxidized when it gives up electrons and reduced when it accepts electrons, just like any other molecule. Each NAD plus molecule is used over and over again. The other one is flavin adenine dinucleotide, or FAD. That also is an oxidation reduction coenzyme, and that is sometimes used instead of NAD+, and that's going to accept 2 electrons and 2 hydrogens to form FADH2 in this process. So the phases of cellular respiration themselves, they include glycolysis, and then there is a preparatory step. Glycolysis is the breakdown of glucose into 2 molecules of pyruvate. Glycolysis means splitting of sugar itself. It does take place in the cytoplasm of the cell. You do generate small amounts of ATP. It does not utilize oxygen, so it is an anaerobic process. Glycolysis occurs both in aerobic and in anaerobic respiration. Before you would enter into any next step there, then you'd have a preparatory step, or the prep reaction, where both those molecules of pyruvate are oxidized and entered into the matrix of the mitochondria. So the preparatory step is part of cellular respiration itself, or true aerobic respiration. Electron energy is stored in NADH, and 2 carbons are released as carbon dioxide, 1 from each pyruvate. So remember, glycolysis, you have 1 sugar going in, and at the end, you end up with 2 pyruvates. So after glycolysis, each reaction is going to take place 2 times. You also have the citric acid cycle. The citric acid cycle, or the Krebs cycle, once referred to, is going to occur in the matrix of the mitochondria and produce NADH and FADH2. The whole purpose of that citric acid cycle is to pump out these electron carrier molecules. It is a series of reactions that does also release carbon dioxide gas, and it turns twice per glucose molecule, hence producing 1 pyruvate at the end of glycolysis. It produces 2 immediate ATP molecules for a glucose molecule as well. But the bulk of the ATP doesn't come out of glycolysis, the prep step, or the citric acid cycle. It's going to come out of the electron transport chain. And the electron transport chain itself is a series of carriers on the cristae of the mitochondria. Remember, the cristae is the foldings of the inner membrane of the mitochondria. And it's going to extract the energy from NADH and FADH2. It's going to pass electrons from higher to lower energy states, and coupled with chemiosmosis, the electron transport chain, as those electrons pass through that membrane or the ETS system, you're going to pump out 32 to 34 ATP molecules using the enzyme ATP synthase, just as we have seen in photosynthesis. So if we look at the 4 phases of complete glucose breakdown, here they are. So you have glycolysis in the cytoplasm, and the electron transport chain and chemiosmosis taking place inside the mitochondria itself. Overall, remember that cellular respiration does occur in both plant and animal cells, but photosynthesis is just strictly to the plant cell, or the cells of those things that are considered photoautotrophs. So overall, a glycolysis, you get out 2 ATP. Then we get out 2 ATP in the citric acid cycle, and then we get out 32 to 34 ATP via the electron transport chain. So outside the mitochondria is when glucose takes place, and depending on what happens, glycolysis, and depending on what happens after glycolysis is going to be determined by oxygen gas availability. So glucose occurs in the cytoplasm. It consists of a series of 10 reactions, each with its own enzyme. Energy investment step is the first part. To get the reaction going, you need to have an input of 2 ATP molecules to activate glucose before glucose is split into 2 G3P molecules. You also then have the second phase of glycolysis, which is the energy harvesting step. And that's going to require the oxidation of G3P by removal of electrons and hydrogen ions. The 2 electrons and the 1 hydrogen ion are going to be accepted by NAD+, resulting in 2 NADH. You also have 4 ATP that are going to be produced by substrate level phosphorylation. Substrate level phosphorylation, remember, is going to use an enzyme to pass a high energy phosphate group to ADP to make that ATP molecule. So ADP becomes phosphorylated. There is a net gain of 2 ATP, 4 AT produced, 2 ATP consumed. Both G3Ps are going to then be converted to pyruvate. So if we look at the inputs and outputs, the inputs are 6 carbon glucose 1, 2 NAD+, 2 ATP, and you get 4 ADP plus 4 phosphate. The outputs are 2 3-carbon pyruvates, 2 NADHs, 2 ADP, and 4 ATP total. So the net gain of ATP here is only going to be 2. If we look at the energy investment step, basically you have glucose. Glucose is going to be phosphorylated to form 1,3-bisphosphoglycerate. Then you get the two ATP molecules used to get started there to form that G3P. G3P is the, if you recall, the end product of what we saw in photosynthesis, glyceraldehyde 3-phosphate. That G3P then is going to go into the energy investment step and be converted to some intermediate molecules. And there we could see the NADH being formed. And here we could see the net gain of ATP there. And ultimately, it keeps going to form more intermediates. Then we get out two more ATP molecules. And ultimately, at the end down here, we do see that we end up with pyruvate, 2 3-carbon molecules of pyruvate. So we have a net gain there of 2 ATP. 2 in, 4 out yields 2 altogether. Substrate level ATP synthesis. Basically, this is an illustration of what is going on there for substrate level cellular respiration. So outside the mitochondria, if oxygen is not available, then we do have fermentation taking place. And pyruvate is a pivotal metabolite in cell respiration. If oxygen is not available to the cell, fermentation, which is an anaerobic process, occurs in the cytoplasm. This is during fermentation, glucose is incompletely metabolized to lactate or to carbon dioxide and alcohol, depending on the organism. If oxygen is available to the cell, pyruvate enters the mitochondria for aerobic respiration. So fermentation is an anaerobic process that reduces pyruvate to either lactate or alcohol, which would be ethanol and carbon dioxide. NADH transfers its electrons to pyruvate. That is a key thing there, the electron transfer there. If you stop yielding NAD plus to go back into glycolysis, then that whole process would come to a halt. So alcoholic fermentation is carried out by yeast. It produces carbon dioxide and ethyl alcohol. It's used in the production of alcoholic spirits and breads. Lactic acid itself is carried out by certain bacteria and fungi. It produces lactic acid, or what we would say is lactate, used commercially in the production of cheese, yogurt, and sauerkraut. Other bacteria produce chemicals anaerobically, including isopropanol, butyric acid, propionic acid, and acetic acid, which is vinegar. So if you look at the overall steps here, here you can see the difference between alcoholic fermentation and lactic acid fermentation, which is essentially right here. So this here is glycolysis taking place, and you end up with pyruvate, but afterwards you, without oxygen gas present, depending on the organism, so in this direction we have animals, each pyruvate would go through and you'd form lactate, or here in plants, you go through and form two alcohols and carbon dioxide. Overall, you only do get that net gain of 2 ATP, and that 2 net gain of ATP is essentially coming out of glycolysis. But what's important here is the NADH that is made in glycolysis is actually coming through and donating the electrons there and regenerating NAD+. And that could come back in and keep picking up those electrons to keep this entire process going in a cyclic manner. So here you see some fermented foods. So outside of the mitochondria, there are advantages to fermentation itself. It does provide a quick burst of ATP energy for muscular activity. When muscles are working vigorously for a short period of time, lactic acid fermentation is going to provide that energy. However, the disadvantage of that is as we have lactic acid fermentation taking place in our cells, it's going to change the pH levels of our cells themselves, and that's going to cause our muscles to experience fatigue due to oxygen debt. And that fatigue or that burning sensation would be the buildup of lactic acid in our muscles. And we do know that lactic acid or lactate will go back to the liver and be converted back into another product. Lactate and alcohol are both toxic to cells. Yeast die from the alcohol they produce by fermentation. So overall, when we look at the efficiency of this process, it's not as great as what we'd see in true aerobic respiration or cellular respiration. You do get 2 ATP produced per glucose during fermentation, and that's an equivalent to 14.6 kilocalories of energy. The complete oxidation of glucose can yield 686 kilocalories. So the efficiency is about 2.1% of the total possible for glucose breakdown. Only 2 ATP per glucose are produced compared to the 36 or 38 ATP molecules that are produced by cellular respiration. So outside the mitochondria, overall, here is the summary chart of inputs and outputs for fermentation. You should know the inputs and outputs for all these processes. So inside the mitochondria, we do have true aerobic respiration taking place because oxygen is available. And before we go from glycolysis into the citric acid cycle, we have this connecting step called the preparatory step. The end products of glycolysis, which is pyruvate, enter into the mitochondrial matrix. Pyruvate is going to be converted into a 2-carbon acetyl group. It's attached to a coenzyme called coenzyme A to form acetyl CoA. The electrons are picked up as a hydrogen atom by NAD+ to produce NADH. And you also release carbon dioxide and transport it out of the mitochondria into the cytoplasm. This occurs twice because at the end result of glycolysis, you have 2 pyruvate molecules. So the 2 pyruvate each will go through this prep step if oxygen is available. And here you can see the overall equation and the molecular structures for those molecules themselves. Ultimately, then, what would happen is inside the mitochondrial matrix, the cristae is the location of the electron transport chain, but the location of the prep step in the citric acid cycle is in the matrix here. So the citric acid cycle would take place following the production of acetyl CoA. This process, also referred to as Krebs cycle, occurs in the matrix. It begins with the addition of 2 carbon acetyl groups to form acetyl CoA to a 4-carbon molecule called oxaloacetate. You heard that word before. It was in one of the alternative pathways of photosynthesis. That's that acid that's formed. It forms a 6-carbon molecule of citric acid, hence the citric acid cycle. So oxaloacetate forms citric acid. You get out NADH and FADH2 capture the energy of rich electrons. ATP is formed by substrate level phosphorylation. This occurs twice for each glucose that is going in, and it produces 4 CO2, 2 ATP, 6 NADH, and 2 FADH2 per glucose molecule. If we look overall then, the cycle begins when the acetyl group carried by the CoA combines with C4 molecule to form that citrate. So oxaloacetate comes in, binds with acetyl CoA, and you form citrate. Ultimately then you get an intermediate there and you get the production of NADH and CO2. And then going from that intermediate to succinate, you get out another NADH and an ATP molecule and carbon dioxide gas again. So from succinate to fumarate, you get out an FADH molecule there, and the cycle repeats itself. From fumarate back to the oxaloacetate, you do get out another NADH molecule. So there you can see 3 NADH and 1 FADH2 and 1 ATP are produced for one turn of the cycle. So overall, our outputs are 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP per glucose molecule. So the fun stuff really occurs at the cristae of the mitochondria, because this is where we're going to pump out a lot of our ATP molecules, because this process is coupled with chemiosmosis. The location in eukaryotes is the cristae of the mitochondria. For aerobic prokaryotes, aerobic bacteria, That's going to be in the plasma membrane of those cells. You have a series of carrier molecules that are going to pass those rich energy electrons successively from one to another. A complex array of proteins and cytochrome complexes. The proteins with heme groups have central iron atoms to them. You could see that in the structure, or the molecular structure of these. So iron is the center part of all these molecular rings. The electron transport chain does take in those electrons from the two electron carriers, NADH and FADH2, and it produces ATP by oxidative phosphorylation. Ultimately, why is oxygen needed in this entire process? It's because oxygen is going to be the final electron acceptor. It's going to combine with hydrogen ions to form a water molecule, which is a byproduct of this entire process. So the fate of the hydrogens from NADH deliver enough energy to make 3 ATPs. Those from FADH2 have enough energy to make 2 ATPs. The spent hydrogens are then combined with oxygen. The recycling of the coenzymes does increase efficiency. Once NADH delivers hydrogens, it returns as NAD plus to pick up more. However, the hydrogens must be combined with oxygen to make water. So if oxygen is not present, NADH cannot release the hydrogen ion, and it is no longer recycled back to that NAD plus molecule. The electron transport chain complexes pump hydrogen ions from the matrix into that intermembrane space of the mitochondria. The hydrogen ions therefore become more concentrated in the intermembrane space. The pH in the intermembrane space would be lower than other parts of the mitochondria. And ultimately, what does that do? It does create an electrochemical gradient of hydrogen ions. And basically what will that then allow is those hydrogen ions will pass through ATP synthase as they flow down the gradient. The flow of hydrogen ions drives the synthesis of ATP from ADP and inorganic phosphate. The process, as we learned in photosynthesis, is the same here. That is chemiosmosis. ATP production is linked to the establishment of that hydrogen ion gradient. ATP moves out of the mitochondria and then can be used for cellular work. Here you see a diagram of what that process looks like. ETS coupled with chemiosmosis. So ultimately, the energy yield from glucose metabolism, the net yield per glucose from glycolysis, it's 2 ATP. The citric acid cycle is 2 ATP. And the electron transport chain yields 32 to 34 ATP molecules. Energy content, reactant, glucose itself is 686 kilocalories. The energy yielded from 36 ATP would be 263 kilocalories. So the efficiency itself is about 39% or 40% respectively. So the rest of the energy from glucose is lost as heat. So with 39 to 40% efficiency, we're going to lose 60 some percent of that energy, or 61% of that energy as heat. So it is not although we do yield more energy out of this process than we do fermentation, it is not really energy efficient. And that could take us back to those laws of thermodynamics that we started to look at the very beginning of our energy unit. So energy yield from glucose metabolism, if we go through and just look at these, here we could see overall what we get out of all this, all these processes together. So here we do have the cytoplasm, and this pinkish area would all represent the mitochondria. So the subtotal would then be 32 or 34 ATP. So overall, we get 36 to 38 ATP total for the entire process of aerobic respiration. So metabolism, ultimately, if we look at foods, it's the source of energy-rich molecules, carbohydrates, fats, and proteins. It's a degradative process. Basically, it's a catabolic reaction, which is going to break down molecules. It tends to be exergonic in nature because it's going to release energy. We looked at energy reaction diagrams and we looked at endergonic versus exergonic. Synthesis reactions and anabolisms build molecules. Overall, those tend to be endergonic and consume energy like we see in photosynthesis. So the metabolic pool concept basically is just illustrated here with pyruvate and how all these things essentially could be converted between proteins, carbohydrates, fats, so on and so forth, and how all these things can also be broken down to produce ATP. When you look at this overall picture, it kind of makes you think about um, like diets that are low sugar diets or low fat diets, but may have a lot of sugar in them, or drinks. How just these foods can be converted from one form to another. Sugars into fats, so on and so forth. When you're looking at your overall this overall metabolic pool concept. So overall, glucose is broken down in cell respiration. Fats break down into glycerol and 3 fatty acids. Amino acids break down into carbon chains and amino groups. That's called deamination. Basically, the NH2, that amino group, is removed. That does occur in the liver. It results in poisonous ammonia, but that can be quickly converted to urea, which we can expel through excretion. Basically, different R groups from the amino acids are processed differently. Fragments do enter respiratory pathways at many different points. All metabolic compounds are part of the metabolic pool. The intermediates from the respiratory pathways can be used for anabolism, which is the synthesis reaction of metabolism. So anabolism, we look at carbohydrates. It starts with that acetyl-CoA and basically reverses glycolysis, but using a different pathway there. Fats, G3P is converted to glycerol. The acetyl groups are connected in pairs to form those fatty acids. Anabolism of proteins, they make up the combination of 20 different amino acids. Some of the acids, 11, can be synthesized by adult humans. However, the other amino acids, 9, cannot be synthesized by humans. Essential amino acids, and those are essential amino acids, and they must be present in the diet. So the energy organelles revisited once again. Similars between photosynthesis and cell respiration is the use of membranes. We do have the electron transport chain. We do have chemiosmosis. We do have enzymes that play a role, and specific reaction cycles, such as the Calvin cycle with enzymes and the citric acid cycle taking place with enzymes as well. So you could see how all that is summarized right here in this little data table, or this little note chart. So flow of energy, basically with any natural system, we do see that overall relationship between autotrophs and heterotrophs, especially photoautotrophs here. The energy flows from the sun through the chloroplast to the carbohydrates and then through the mitochondria and into ATP molecules. So you can see essentially how important photoautotrophs are for all living creatures on our planet, except for those in the ocean deep that would rely on chemosynthesis. So that's it for our lecture. That covers the entire chapter in about 30 minutes. Thank you and have a nice day.